Welcome back, fellow mitochondriacs, to another episode of Cancer as a Mitochondrial Metabolic Disease Basics. Cancer cells love sugar. This is not an internet myth. This is built on over 100 years of biochemical research and discovery. Not because sugar causes every cancer, although hyperglycemia or high blood sugar and insulin can clearly contribute, but because most, if not all, cancer cells are metabolically rewired to pull in massive amounts of glucose and rapidly convert it into lactate. This abnormal sugar burning program is called the Warburg effect. But this is where it gets interesting. EGCG, the main catechin in green tea, appears to interfere with several parts of this glucose metabolism system. It appears to affect glucose transporters, glycolytic enzymes, lactate production, and signaling pathways that help cancer cells maintain this high glucose state. So in this video, we're going to ask a very specific question. Can EGCG help starve cancer cells by disrupting the Warburg effect? We shall soon see. So without further ado, let's get into it. So as I alluded to in the intro, we are going to talk about EGCG, the flavonoid catechin, as part of the green tea plant, about how it can affect glucose metabolism. And I felt it was very fitting to be sipping on a keto matcha latte during this episode while I was preparing, as you can imagine. So let's get into the nitty-gritty of how EGCG potentially can help us. I'm going to start with a general overview that was in a paper that was published in March of 2024. It's titled Targeting Cancer Hallmarks with Epigallocatechin Gallate, EGCG, Mechanistic Basis and Therapeutic Targets. And in the abstract, it says EGCG is a catechin, which is a type of flavonoid found in high concentrations in green tea. EGCG has been extensively studied for its potential health benefits, particularly in cancer. EGCG has been found to exhibit antiproliferative, anti-angiogenic, and pro-apoptotic effects in multiple cancer cell lines and animal models. EGCG has demonstrated the ability to interrupt various signaling pathways associated with cellular proliferation and division in different cancer types. EGCG anti-cancer activity is mediated by interfering with various cancer hallmarks. This is important to note because when we're dealing with cancer hallmarks, these are generalities that are seen in most cancers. So it kind of skips the idea that does this work in breast cancer? Does this work in prostate cancer? Does it work in GBM? It's going to very likely have these effects in all cancers because these hallmarks are generally seen in most cancers. The studies discussed in this review enrich the understanding of EGCG's potential as a therapeutic tool against cancer, offering a substantial foundation for scientists and medical experts to advance scientific and clinical investigations regarding EGCG's possibility as a potential anti-cancer treatment. And I'm going to, as usual in most of these videos, focus us back on what truly matters when it comes to affecting these pathways. Because we could play biologic whack-a-mole with some of these signaling cascades, but if we don't affect the heart of the engine that cancer runs on, glucose and glutamine, we're really kind of spinning our wheels. So I'm going to focus this particular lecture on the left side of the graphic on glucose uptake, utilization, and the creation of lactic acid, also known as cytosolic substrate level phosphorylation, or the Warburg effect. So later in the paper, it says the role of EGCG in tumor dysregulated metabolism. EGCG has emerged as a promising candidate for targeting tumor dysregulated metabolism. Its ability to modulate various metabolic pathways involved in energy production, lipid metabolism, and redox balance highlights its potential as a therapeutic agent for combating cancer. Tumor cells tend to rely on glycolysis, or the burning of glucose, for energy production, even in the presence of oxygen. That's the Warburg effect. EGCG has been found to inhibit key enzymes involved in glycolysis, such as hexokinase and pyruvate kinase, thereby disrupting the Warburg effect and inhibiting tumor cell growth. It was noticed that a combination of EGCG and gemcitabine reduced pancreatic cancer cell growth in a xenograft model by 67%, which was explained by suppressing glycolysis. Plus, in colorectal cancer, EGCG treatment of cancer-associated fibroblasts restrained the tumor-promoting abilities by stopping their glycolytic activities. Also, EGCG has been shown to inhibit fatty acid synthase, an enzyme involved in de novo fatty acid synthesis. By suppressing lipid synthesis, EGCG can interfere with the formation of lipid membranes essential for tumor cell proliferation. Again, raw materials, not fuel. Additionally, EGCG can activate AMPK, a metabolic regulator that inhibits lipid biosynthesis and promotes fatty acid oxidation. In the literature, it was informed that EGCG sensitizes radioresistance by reducing fatty acid synthase, and EGCG acts as a radiosensitizer for better treatment of nasopharyngeal carcinoma. Moreover, in triple-negative breast cancer, it was stated that EGCG inhibited fatty acid synthase. Again, these are hallmarks seen in multiple cancers, which is why you're seeing so many diverse cancers mentioned in just a little tiny blurb of the article. And in this text, it talks about how EGCG is inhibiting key glycolytic enzymes. And it mentioned specifically hexokinase and pyruvate kinase. And in this particular graphic, specifically having an effect on PFK, phosphofructokinase, one of the glycolytic enzymes. I then pulled a paper that specifically looked at EGCG's ability to inhibit glycolytic enzymes at varying concentrations. And so what we have up on the top left part of the graphic and the top kind of middle part of the graphic are the mRNA expressions, or the amount of proteins that are being made de novo, and how either normal control or under the influence of 100 micromoles of EGCG. And you see here that there's not a lot of, or really any activity at the level of GLUT1, even under 100 micromoles, but there is at GLUT2, there is some at GLUT3, there's some at GLUT4. Hexokinase, there's a decrease here, but PFK seems to be, of all the targets that EGCG has, PFK seems to be the most vulnerable to EGCG in this particular chain of events of glycolysis and glucose metabolism. And what they did was, in these other graphics, they actually looked at PFK activity at varying concentrations. So here they were looking at giving 100 micromoles. At this one, they were giving like increasing doses of EGCG from 0 to upwards of 250 micromoles. And you'll see here that at 50 micromoles, you're seeing that there is a roughly 50 to 60% decrease at those levels. And then in another cell line, it looks like at 50 micromoles, you were having about a probably 40% decrease activity at 50 micromoles. And then I actually had AI look at these graphics and actually give me better estimations that I can make from my naked eye here about the activity. So at 5, it was 78 to 85, and probably be better to look at it from this perspective. There's about 15 to 22% reduction. At 10 micromoles, there's 28 to 35% reduction. At 20 micromoles, it's about a 42 to 50% reduction. And at 30 micromoles, it's about 52 to 60% reduction in PFK activity. And again, this is not directly inhibiting this enzyme. What it's doing is it's blocking the cell's ability to actually make this enzyme via lowering gene expression. And that's shown by these mRNA expression studies here at the top. So pretty astounding. So I pulled a graphic that was from the same paper up here, and it was basically showing all the different studies that were done in vitro and vivo for different types of cancer using EGCG, looking specifically at tumor dysregulated metabolism. And there's several studies. Most of these probably in vivo studies are very likely to be preclinical, not human or clinical trials in humans. In the conclusion, it says cancer continues to be a leading cause of mortality and morbidity worldwide, despite significant recent advancements. Anti-cancer medications are successful in treating cancer. That's debatable, but they can have negative effects. That's probably not debatable, such as alterations in physiologic and biochemical processes, exhaustion, hair loss, infection, nausea, diarrhea, etc. The evaluation of numerous biological compounds and natural products has been demonstrated to have a key role in prevention and suppression of cancer. The prevalent catechin in tea, epicatechin 3 gallate, and its significance in healthcare and illness prevention have been documented. Many experimental and clinical research studies have also noted that EGCG has anti-cancer properties. It can be used alone or in conjunction with other treatments to accomplish its inhibitory effects. Metabolic therapies are meant to be used in conjunction as a team, not as a unilateral intervention. EGCG is a key physiologic anti-cancer drug due to its role in triggering multiple anti-cancer processes in many cancer hallmarks. To consider EGCG as a routine therapeutic option to treat various malignancies, more clinical studies are required. And that is probably the most common end phrase of all of these studies on these very prominent and exciting compounds, whether it be a natural compound like EGCG or something else like mebendazole or ivermectin. It'll say, well, even though there's tons of mechanistic data here, it's not ready for prime time. We need more clinical studies, more clinical studies. And then unfortunately, those clinical studies never get done. So we're left with what we have, which is a lot of times in vitro or preclinical models, which is really unfortunate. The next paper I'm going to pull from was published in January of 2024. It's titled The Roles of Epicatechin Gallate in the Tumor Microenvironment, Metabolic Reprogramming, and Immunotherapy. And I stole from the abstract here. It says, additionally, EGCG can suppress multiple metabolic reprogramming pathways, including glucose uptake, aerobic glycolysis, or the Warburg effect, or cytosolic substrate level phosphorylation, glutamine metabolism, fatty acid anabolism, or the creation of new fatty acids, and nucleotide synthesis. And this paper had a pretty awesome, but also very busy graphic. And basically everywhere you see this, green shape with EGCG in the middle, that's where EGCG is having an effect. We are specifically in this video looking at the left side of the screen. We're looking at the glycolysis system. We see glucose uptake channels, hexokinase, PFK, PGAM, PKM2, lactate dehydrogenase A, G6PD, et cetera, et cetera, et cetera. And then there's a whole bunch of other metabolic pathways that are also inhibited or affected by EGCG. It's just that we're focusing our attention on this side of the graphic for this particular video. It's unbelievable, truly, what EGCG is capable of. Regulatory roles in tumor metabolic reprogramming. Tumor metabolic reprogramming provides a constant supplement to tumor energy consumption and biosynthesis to meet uncontrolled proliferation, which concurrently provides critical targets for tumor therapy. To further elucidate EGCG's suppressive effects on tumor cell metabolism, we summarized the regulatory roles of EGCG in multiple upregulated metabolic pathways, including glucose uptake, aerobic glycolysis, glutamine metabolism, fatty acid anabolism, and nucleotide synthesis. And I highlighted or snipped out the portions that we're talking about today on glucose metabolism. The metabolic flux of glucose depends on glucose uptake by tumor cells, during which glucose enters tumor cells via active transport. On the cell membrane of tumor cells, glucose transporters, GLUT, proteins are often upregulated to promote glucose uptake and support tumor metabolism. Upregulated GLUT1 levels have been confirmed to be an important indicator of poor prognosis in multiple cancers, including lung, pancreatic, colorectal, ovarian, breast, bladder, esophageal, and oral cancers. Basically every cancer, because this is seen in every cancer as a cancer hallmark. Therefore, targeting GLUT offers excellent prospects for anti-tumor therapy. A host of experimental findings shed light on the targeted inhibitory roles of EGCG in GLUT-dependent glucose uptake in tumors. Following exposure of colon cancer cells to different EGCG dosages, a sustained reduction in GLUT1 mRNA levels was detected. This inhibitory effect appears to be closely correlated with AMPK signaling after EGCG treatment. Similarly, EGCG was observed to reduce GLUT1 expression in breast cancer and pancreatic cancer, although mechanism may be attributed to its effect on HIF1 alpha or MYC or what are the other signaling pathways, AKT, PI3K, etc., which is vital for regulating the genes encoding GLUT1 and ameliorating the effects of glycolytic enzymes. As far as we know, EGCG's effect on HIF1 may partially result from suppression of upstream insulin receptor, IR, and insulin-like growth factor 1 receptors by EGCG. So this just goes to show that insulin-like growth factor are keys at upregulating important growth signaling pathways, which then interact with HIF1. And it just goes to show also that having a ketogenic diet or a low insulin diet is also a very important piece of this that cannot be ignored. In addition, EGCG was discovered to limit GLUT-mediated glucose uptake through directly and competitively inhibiting GLUT1 in breast cancer cells. So there's two ways of affecting these transporters. You can either downregulate their expression, effectively not allow the cell to make the transporter, or you can directly inhibit it. And up until now, we've only seen that it's decreasing mRNA expression of these transporters. And what it's saying here is there's actually direct evidence that EGCG can directly inhibit GLUT1 in breast cancer cells, which is pretty awesome. Relevant molecular docking also supported the direct interaction of EGCG with GLUT docking sites at the core of GLUT1 through hydrogen bonding and hydrophobic contacts. Briefly, EGCG can inhibit GLUT1 expression and target GLUT1 activity to interfere with glucose metabolism in tumor cells. And I like that there's multiple mechanisms even on the same transporter. It's pretty awesome. Aerobic glycolysis. Normal cells obtain energy through oxidative phosphorylation. Why? Because they have functional mitochondria and they have oxygen. Cancer has neither of those. While most solid tumors and non-solid tumors depend mainly on glycolysis to generate energy to adapt to their TME even when oxygen is abundant, a phenomenon named the Warburg effect. This pattern of energy supply seems uneconomical on the surface, but it is essential for tumor cells because it provides them with a survival advantage and makes their TME more favorable for tumor progression. That's the lactate and the succinic acid that is the byproduct of glucose and glutamine metabolism, which sets up an important piece of this TME and acts as kind of like a shielding system from our immune system and from chemotherapy, etc. Hence, targeting the Warburg effect of tumor cells is an extremely promising treatment approach. This means that the Warburg effect, many crucial regulatory enzymes, including hexokinase, phosphofructokinase, PFK, phosphoglycerate mutase 1, PGAM1, pyruvate kinase, PKM2, and LDH or lactate dehydrogenase can be considered as potential targets for antitumor drug development. It has been reported that a novel biological orthogonal probe was used to identify the direct target protein of EGCG based on in situ global proteomics profiling, and subsequent gene ontology analysis revealed that the main targets belong to enzymes regulating pivotal metabolic processes such as glycolysis and energy homeostasis. This investigation declared a unique EGCG interactome and provided an important reference for EGCG to regulate the metabolic process of glycolysis, or the burning of glucose. Hexokinase can catalyze the phosphorylation of hexose's 6-hydroxyl group to generate glucose-6-phosphate, the first rate-limiting stage in glycolysis. Preclinical experiments confirm that EGCGs dose-dependently suppress the activity and mRNA levels of hexokinase 2, reduce glucose consumption, and impaired lactate and adenosine triphosphate production, ATP production, and thus the tumor growth was remarkably inhibited. Related evidence further pointed EGCG could significantly downregulate hexokinase 2 expression through strongly inhibiting epidermal growth factor EGFR activation and downstream AKT phosphorylation, thus exerting its far-reaching antitumor effects through the direct regulation of glycolysis. Molecular docking and dynamics simulation analysis also identified EGCG with obvious binding preference and selectivity for the HK2 binding pocket. This is even more astounding. So not only does it downregulate the expression of GLUT transporters and of hexokinase, it directly interacts with GLUT transporters and hexokinase, which is the rate-limiting enzyme in glycolysis, and it attaches to itself and it blocks it. And it could potentially attach to the corresponding active site to form a stable protein ligand complex. Taken together, these results imply EGCG as a potential hexokinase 2 or HK2 inhibitor can potentially inhibit the initiation process of glycolysis. Astounding. This is why EGCG is one of my favorite flavonoids and natural compounds to study of all time. And I'm going to show you why. Let's take a look at all the levels of glycolysis and where EGCG has been shown to affect GLUT transporters. We spent a decent amount of time talking about GLUT transporters. GLUT1, GLUT2, GLUT5, SLGT1, GLUT4, hexokinase, PFK. We've talked about all of these so far. PFKFB, maybe. PKM2, we've talked about that so far. LDHA or lactate dehydrogenase A, MCT transporters or lactate transporters. And it's been mentioned in passing, but it's also an inhibitor or a destabilization factor for HIF1 alpha. So if we were to look at where EGCG is acting on glycolysis and the metabolism of glucose by cancer cells, I have put an arrow and the word EGCG by every area that it's been shown to affect. So glucose transport, hexokinase, HIF1 alpha, PKM2, PFK, PKFB, lactate dehydrogenase A, and lactate transporters or MCT transporters. It's just astounding what these natural compounds can truly do. And this is just looking at glucose. If we look further down, again, here's EGCG, again, working at the level of fatty acid synthesis, cancer-related signaling pathways, such as AMPK, PI3K, AKT, mTOR, STAT3, c-MYC, et cetera, as well as reduction in tumor-related inflammation. And that's not all, because in the next video, I'm going to talk about how EGCG can target glutamine metabolism as well, and it may be a direct competitor to the more famous glutamine inhibitor, DAWN. So if you like videos like this, please like, share, subscribe, hype, and until next time.